Good morning. I'd like to, first of all, thank Juliana and all the organizers for this wonderful meeting in Florence. Your tenacity and your organizational skills have been wonderful. We've enjoyed being here, meeting with all of you last evening, and thank you for making this meeting possible and come to be. I also would like to take this opportunity to thank Dr. Sweeto, who I've worked with for since 1998, 99, and we have worked together and spoken together for all these years. And of course, it's wonderful to come together at this meeting and tell you that I believe we've made great progress, even though it may seem like it has been a struggle. I believe that things are changing and that people do or have, through the efforts of Dr. Sweeto and many of us that have served on the Pandas consensus group in the United States and throughout the world, that people are beginning to understand the nature of pandas. I have also got some conflicts of interest to declare and thanks to give. My trip here was supported by my laboratory, Molecular Labs in Oklahoma City, and also by my European counterpart, WESLab, and Dr. Michael Ako, if he would raise his hand, is in the back. And I thank them for making it possible for me to come to this meeting and give you this lecture. I also would like to recognize, I don't know if she's in the audience, Dr. Graziella Orefici. If she's here, please raise your hand. I haven't seen her oh, there's Graziella, sorry. Maybe I'm still asleep. But Dr. Orefici has been a supporter, certainly, of Group A streptococcal sequelae and diseases for her entire life. And she has brought much international recognition to Italy because for, 15 years, she has brought the international meeting of streptococci to Italy, to Sicily, and she is going to be president in Argentina this year. So she has spent her whole life bringing the understanding of streptococcal diseases, and of course now is here at the Pandas meeting with us. And I certainly want to recognize her for all the work that she has done. So for a little bit of history, I've spent my entire life investigating autoimmunity in rheumatic fever. And so how did I get involved in pandas? Well, Dr. Sweeto called me in 1998 and asked, could I help her? Because I had spent 15 years working on rheumatic heart disease. And we believe in translational medicine in my laboratory, and that's trying to understand how the body actually is responding in diseases where we have infections. And so this particular case, as you've heard from Dr. Sweeto, is a streptococcal infection. And I'm going to have to reach over here. I'm going to take the microphone off. I think that will make it a little bit easier for me. The streptococcal infection, as Dr. Sweeto has mentioned, has a number of manifestations as a disease. It may manifest as pharyngitis or impetigo. In more serious diseases that you've heard about, cellulitis, necrotizing fasciitis, it may present in children as scarlet fever with the scarlatinal rash, and that is due to the erythrogenic toxin expression by the group A streptococcus that causes the red rash. And the strawberry tongue, always make them stick their tongues out. Septicemia and acute glomerulonephritis. Septicemia, of course, bacteria in the blood, and acute glomerulonephritis, the kidney manifestation of post-streptococcal disease. But acute rheumatic fever has been the subject of my laboratory for really 33 years. And Dr. Sweeto mentioned molecular mimicry. Of course, this is the sharing of antigenic determinants between a host and invading microorganism. And to give you a little better understanding, for all these years we've investigated various components of the Streptococcus, and when you go and think you have a strep throat, you run to the clinic and they do a quick strep test, and they're identifying the strep based on its group A carbohydrate. Well, that's one of the antigens that actually is responsible for the antibodies that actually bind to the basal ganglia. The streptococcal M protein is also well known to mimic alpha helical coil molecules, and I think we're probably more well known for our work in rheumatic heart disease, where the cardiac myosin is very similar to M protein, but it also mimics the group A carbohydrate, because we have begun to understand that antibodies don't always recognize one single antigen. And that's what mimicry is about, is that their antigens are components on the streptococcus that look like components in the host. And so antibodies are misdirected or attack the host components when really they were intended for the group A streptococcus. And of course, long after the streptococcus is gone, that's when you actually see disease. That's why it's called post-streptococcal. And in fact, on the website of one of our Denmark colleagues from the Staten Serum Institute, he calls pandas post-streptococcal autoimmune encephalitis. The autoimmune manifestations of acute rheumatic fever, and Dr. Sweeto briefly mentioned these, but of course, polymigrating arthritis, which is the most common manifestation, and the most serious, of course, is the valvular heart disease that we call rheumatic carditis, that can, of course, last a lifetime and result in valvular replacements. But today, I'll be more interested in bringing to you our work on Sydenham chorea with the CNS manifestations, and then, of course, erythema marginatum and subcutaneous nodules are well recognized as a rash, and then, of course, the subcutaneous manifestation of rheumatic fever. Oh, I hit the wrong computer. This is a case of Sydenham chorea. It's a 12-year-old young lady. And she is asked to put her hands out. And she's asked to also touch her nose in a moment. And you'll see that she will not be able to do that. And these involuntary movements, of course, incapacitate children. And so the streptococcal-associated behavior and movement disorders fall into now two categories, because we've identified something new, pandas, that are PANS that comes along that may be related to Sydenham chorea with the early neuropsychiatric symptoms that follow, that are followed by these severe disruption of movements. And of course, for parents, when you see a child and they have Sydenham chorea, you're so worried about their movements that you're not even thinking about the neuropsychiatric problems. So we kind of forgot about this for a while. And I began to collaborate with an individual in Israel, Dr. Hilla Ben-Pazi. She's here. She has a neurology meeting. She was here earlier today. And I'll talk about her work toward the end of my presentation. But she actually helped me as we went through her data to understand how the neuropsychiatric symptoms may be present at the beginning. And of course, pandas and PANS, you've heard lots about that from Dr. Sweeto. And of course, we're asking the question, is Sydenham chorea a streptococcal-induced autoimmune sequelae? Is it a path to understanding neuropsychiatric disorders associated with autoimmunity and infection, really not only in children, but perhaps even in the adult populations? And our translational approach, because we believe in translational medicine, is to study the mimicry and the autoimmune streptococcal sequelae using human monoclonal antibodies, where we take blood from children and we take a B cell that actually makes the antibody and we fuse it with a malignant cell that makes this B cell live forever. And then we have a reagent for which we've studied rheumatic heart disease for over 30 years. These reagents actually last in the laboratory, and we can get these hybridomas out, B-cell hybridomas, and study the antibodies for a very long time. And we can identify brain and streptococcal antigens and also study the pathogenesis of the disease, because when we first began the study, we studied the antigens. But then we began to realize that we could actually study the pathogenesis of the disease using these antibodies. So the mimicry, to summarize quite a number of years of work, the mimicry in Sydenham, Korea comes down to cross-reactivity between antibodies with the group A carbohydrate, which has the epitope N-acetyl beta-D-glucosamine, and lysoganglioside and tubulin. In the case of rheumatic heart disease, the antigen is cardiac myosin and other antigens like laminin that are on the surface of the valve. In the case of the brain, lysoganglioside and tubulin are very highly enriched in the brain tissue. And so these were the first antigens that we identified in disease. And this is showing 3 of the human monoclonal antibodies, actually from an Oklahoma patient that I saw in the hospital. And we obtained her blood, and we produced these 3 monoclonal antibodies and studied them and determined that they reacted very strongly with lysoganglioside, whereas another antibody did not. That was a control. And just to prove that it's not Guillain-Barré syndrome or some other type of nerve disorder, we actually studied all the gangliosides that were studied in Guillain-Barré. And you can see that these were all negative, and negative means that it's greater than 500, because we were doing an inhibition, which for immunologists is really the ultimate. We want to prove that an antibody reacts with an antigen, and of course the easiest way to do this is to mix it with the antigen and do an inhibition. So these are actual concentrations of inhibitor. But the point is that lysoganglioside stood out, and look at monoclonal antibody 24, because it would be the key to Sydenham, Korea. We also demonstrated, because you don't want to think that one single monoclonal antibody reflects the entire disease. And so what we did was Dr. Sweeto came to our rescue and said, here are all these Sydenham, Korea patients that we had at the National Institute of Mental Health. And we took these 3 that we had matched sera, both acute and convalescent, and showed that the anti-lysoganglioside antibodies were elevated, and in convalescence they were greatly reduced. And so when we began to look at larger numbers and look at the Pandas patients that Dr. Sweeto had provided us, we found that very interestingly, they had high levels. My pointer's not working, I think. Oh, there it is. High levels of the anti-lysoganglioside antibody, very significant. And notice the little Tourette syndrome group that actually we had a significant level, whereas in the ones with Tourette's that had OCD did not, nor the ADHD or normals. We actually think that eventually we will show that the anti-lysoganglioside probably has a tendency to be present more in ticks. And Dr. Sweeto showed you this slide, but we'll just review it again, that with Sydenham, Korea, and Pandas cerebrospinal fluid, because cerebrospinal fluid is very important. This is where the antibody is going to concentrate and get into the brain. And this will be IgG, and it was shown to be prominent in the basal ganglia, and probably this differential staining is a nerve tract in the basal ganglia. We then didn't sit still at identifying the antigens. We decided to ask the question, what about the mechanism of action of these antibodies? Could they actually cause a problem in patients? And the reason we tested them for cytotoxicity and found that they were not particularly cytotoxic. And we began to ask the question, you know, patients with Sydenham chorea, do they die? No, they don't die. They actually get better eventually, and they don't seem to have major neurologic deficits. And so we wanted to ask, are they functioning by signaling? And so we looked at whole cells and took our monoclonal first and another anti-ganglioside antibody, and noticed that they bound in lumps. They had big blobs of antibody on the surface of the cells. And the antibody from Sydenham chorea looked very much like a commercial anti-ganglioside antibody, whereas the control did not bind. And so we did find that the monoclonal antibody and chorea sera, because you always go back to the patients, you don't depend on a monoclonal antibody to answer all your questions, and studied the cell signaling through calcium calmodulin-dependent protein kinase 2. And this happens by taking the neuronal cells, and we have a human neuronal cell line, you mix it with the antibody, and then you look to see if something went crazy inside the cell, and that happened to be the CAM, kinase 2. And weren't we lucky, because we found that monoclonal antibody 24 signaled. What if we hadn't had monoclonal antibody 24? We were very, very fortunate. The other two antibodies did not signal similarly, and was similar to controls and the basal CAM kinase level in the cell. And you can see also the donor that I saw in the person in the hospital, their sera signaled, and so we were very excited and took the sera after she had gotten better and the signaling was gone. So we were even more excited, and so was Dr. Sweeto, and so she provided even more acute sera from chorea and convalescent sera, and we showed that they too had the signaling, the CAM kinase signaling of the human neuronal cells, and the convalescent sera did not. And so what does this mean? What is the real mechanism? And so we used tritiated dopamine in a test where we mixed the antibody with the cells and looked for tritiated dopamine release, because CAM kinase is an enzyme that is in the cell and eventually helps make dopamine. And so what the patients were doing was binding their cells and making too much dopamine. And so that's what this shows. The big, tall red bars are the elevated dopamine release by antibody, by either chorea sera or by the monoclonal antibody. And so Dr. Sweeto showed you this slide. This is a culmination of studies of non-Pandas sera, Pandas sera, and Sydenham chorea sera. And you can see the signaling of CAM kinase is very strong in Sydenham chorea. It overlaps with the Pandas group, but the normals and the non-Pandas are much lower. And so it appears that it may be a biomarker for these diseases. And so the diagram that we've had for some time, the Sydenham chorea autoantibody would bind to the surface of a neuronal cell, trigger CAM kinase, trigger tyrosine hydroxylase, which identifies dopaminergic neurons, and induce dopamine release and lead to an antibody-mediated imbalance in the dopamine, central dopamine. So we decided to put this antibody into rats, and we did intrathecal administration to see if we could get elevated tyrosine hydroxylase, and in fact we did, to prove that in fact the antibody drives the pathway on from CAM kinase to tyrosine hydroxylase, which then goes on to make too much dopamine. So we then wanted to show that the antibody would get into the brain and that it would target dopaminergic neurons. And so we spent 9 years creating a transgenic mouse to express that one human monoclonal antibody from the patient with Sydenham chorea. And this is what happened in the basal ganglia. You can see that the antibody in green, we have a marker on the antibody so that it is recognized and you don't see the other antibodies in the mouse. And this is the human antibody that we have treated the animals so that we've broken the blood-brain barrier, and the antibody has gotten into the brain and targeted dopaminergic neurons. And these are stained with tyrosine hydroxylase, and this is merged, and that's really all that you need to see. The controls are all negative. And there were also a few in the cortex, but not in the hippocampus at all. So it targets primarily the basal ganglia. We think that it probably is the substantia nigra in our particular animal. So then, is there evidence for antibodies against dopamine receptors in Sydenham chorea in pandas? Because we began to think about the CAM kinase and also the targeting of the dopaminergic neurons, and wanted to ask the questions about dopamine receptors that were present on the surface of neuronal cells that would react with and respond to these antibodies as if they were neurotransmitters such as dopamine. And I can tell you that our antibodies do, you will see that the monoclonal reacts with D2, and I'm going to move ahead a little more quickly because I don't want to overshoot my time. But then we can see that the pandas and the Sydenham chorea both highly significant in the anti-D2. Other forms of tics and OCD that may have antibodies don't have the D2 antibody, and so it's very easy to distinguish between them. In Tourette's, you don't see any significance, and all these normal patients are negative. So we believe the D2 goes along with the movement in Sydenham chorea and also along with the choreiform movements that are seen in the pandas-like illness, in the pandas illness that was discovered and reported on by Dr. Sweeto. Further proof, I won't go into this in great detail, but we use peptides from D2 to actually inhibit the antibody. This is just further proof, and it bound the dopamine D2 receptor on flag-tagged transfected cells. These are just more science evidence. And the peptide that actually sees the antibody is not in the dopamine binding pocket, so it binds at a different site on the receptor. The antibody, human monoclonal antibody, signals D2. We actually put the gene for the dopamine D2 receptor in transfected cells so that the transfected cells could then be used to prove that the antibodies actually react with the D2 receptor. And it's important, and the reason is because it's a membrane receptor, and you have to be sure that it's really signaling the D2. And so this is just all the evidence that the chorea sera signals. I'm going to go through this quickly. I love the dopamine, the dose response of the antibody, because notice it's so powerful. It acts in such small amounts. Nanogram amounts of antibody are all that are needed. So you may not have to have such hugely high levels in a patient. It can be nanograms that will be signaling. And it also is seen in pandas. This is Sydenham. Maybe the other was No, that was the monoclonal. Then Sydenham chorea signaling, and then Pandas sera signaling. I don't want to belabor this. And then antibody recognition of the dopamine D1 receptor. We think this is important, and actually my work with Dr. Hilla bin Pazi in Jerusalem indicated, and I'll be flying through that at the end also, that the ratio of D1 and D2 antibodies actually correlates with the symptoms of Sydenham chorea. And so we just show here that the levels of D2 and also of D1 are significant. And notice that in Tourette's and Tourette's OCD that we get a significant level of D1, not of D2. And so this is actually what we think will distinguish these two groups of children. Again, the dopamine receptor antibodies significant. You can see when we separate the OCD out that the D1 may actually track with the OCD. And there are peptides of D1 that are recognized by a monoclonal antibody. We also went back to making monoclonal antibodies from patients with pandas, and you can see that the D1 peptide reacts with this particular monoclonal antibody, but not the D2 peptide, so it was highly specific. And this peptide is actually in the first extracellular loop of D1. And these are recognized, this peptide is recognized by Pandas patients. And interestingly, the mechanism of action for this antibody is to make the receptor, actually, I'm going to just move ahead. What happens is the antibody makes the receptor about 50 times more sensitive to dopamine. So you can imagine what happens in a person that has both of these antibodies. They get an antibody to D2, they make too much dopamine. Then they have the antibody to D1. Well, that's making the receptor even more sensitive to all this extra dopamine. So naturally, they're going to make an abnormal behavioral response. So back to the question of is Sydenham chorea a path to understanding neuropsychiatric disorders? And work with Dr. bin Pazi has shown that with the D1 receptor elevated early and the D2 receptor elevated late, potentially this is one of our hypotheses. You would get the early neuropsychiatric symptoms in Sydenham chorea, and we see that D1 is elevated first and the D2 is elevated next. And so when we look at the ratio of these antibodies, anti-D1 and anti-D2 in these patients with Sydenham chorea were both significant. And here's the correlation graph with the symptoms showing that the ratio actually correlates with the symptoms. And I wanted to briefly mention the animal model with Dr. Joel and Dr. Brimberg, where animals were immunized with group A streptococci. I think this model is very powerful because it helps fulfill Koch's postulates, where you take the infectious agent, you give it, and of course this is not live strep, but it is the streptococcus components. And they're immunized the animal, and interestingly enough, the animals could not manipulate their food. And this is what you see in pandas. They can't, their handwriting deteriorates. And in Sydenham chorea also, they have lose some fine motor control. They can't walk a beam very well. You can see the loss of that. And they also go crazy over being groomed. We sprayed them with water and they constantly groom. So this is a wonderful animal model. Treatment with haloperidol, actually, or paroxetine, stops, I'm not going to go through all these graphs, but they stop the symptoms. And the antibodies are deposited again in the basal ganglia and are not deposited in the hippocampus or the cerebellum. And CAM kinase is associated with this And anti-IgG beads removes antibody, and that removes the signaling of the CAM kinase, and the antibodies in this model recognize D1 and D2. So it's really a perfect model to go along with the human disease. And then finally, the autoantibody effects in the brain, the antibodies binding, releasing too much dopamine, sensitizing the receptors, particularly D1, to dopamine, and then of course antibody-mediated increases in central dopamine levels may cause selective activation of dopamine D2 receptors, sensitize D1, and of course lead to the symptoms. In addition, when you continually signal CAM kinase, things do not stay stagnant in the brain. The dendrites on the neuronal cells upregulate various things, one of them being the NMDA receptor, and this may actually lead to more OCD. I mean, we have not tested this model, but it has been investigated, not in pandas or any of these antibodies, but that the continual signaling of CAM kinase is not a good thing. I mean, because you simply lead to other things coming up in the child, in the brain. So with that, I would like to acknowledge all the people in my laboratory, Dr. Sweeto and her group at NIH, people all over Yale University, Dr. Leckman, Tel Aviv, Dr. Joel, Dr. Ben-Pazi in Jerusalem, people in Minnesota, Dr. Kaplan. Even I work with Dr. Singer, and we have a paper that's going out for review on more chronic tics and OCD and the antibodies to lysoganglioside and D1 that are present in these children, and they signal CAM, just are amazing. It doesn't go up and down. The antibodies are already elevated at the beginning because they've been sick already for a year or 2 years or 3 years, 4 years. We thank Dr. Michael Janicki, who without his support, we would not have our 1,000-patient study in my laboratory over a 2-year period. And then, of course, acknowledging all the funding agencies that made it possible for our laboratory to function and be open all these years, the National Heart, Lung, and Blood Institute, the NIMH with Dr. Sweeto, the Oklahoma Center for Advancement of Science and Technology, PepsiCo, Harvard University, OU, Lyme Disease Association, Autism Speaks. We continually hold a Trailblazer Award from them, and Grifols, and then Molecular Labs, my conflict of interest. Thank you.
Bene. Sì. Un sincero ringraziamento alla professoressa Cunningham perché chiaramente con la sua relazione ci ha dimostrato l'importanza
Cunningham, with her presentation, she showed us how important the studies have been. We truly thank Dr. Cunningham heartfully, because with her presentation she proved with in vivo studies how these mechanisms work, but also most importantly we saw the correlation and correlated the symptoms. It is very interesting what we saw about the mice study. We saw that, I've read the study that you mentioned. I think that the mice change their ordinary walking route, and their attitude changed, and they changed their whole behaviors, as far as I remember, because I read the article. Certainly, these studies represent a breakthrough for the future of these disorders, these symptoms. I am certain that there will be questions coming from the audience.
Bene, prego.
Thank you, this was wonderful. My name is Francesca Fusco and I'm a neurologist from Rome. Do you want Italian or English?
In italiano.
In Italian, sorry, I'm going to have to do it.
I have two quick questions. The first is, have you observed in the CSF with children with pandas some BDF interaction? And the other question is whether the mice model can be used both for pandas and the Sydney Macaria.
Ok. Professoressa, siccome non sente l'inglese, allora forse glielo posso dire in inglese. Ok, let me repeat this in English. Okay. Take your good. Oh, she's now doing it? Okay, so two things. One is if the levels of BDNF, brain-derived neurotrophic factor, have been measured in the CSF of patients with pandas, because I'm thinking BDNF because it is the one neurotrophic factor that's specific to the basal ganglia, and it's very decreased in Huntington's disease and in neurological disorders they have. So I'm thinking maybe there is some, one of the mechanisms could be that striatum doesn't make enough BDNF for sustaining an insult to the striatum. One is, and the other question is more basic, and it's about the model, the mouse model, if it can be used for pandas and for Sydenham choreas pretty much in the same fashion.
Yes, I don't know. Hello? Okay. The rat, it's actually a Lewis rat model,
right.
and that's fine. It's good for me to actually air that the Lewis rat model we have used for many years as a rheumatic heart disease model, where we actually have created rheumatic heart disease in the Lewis rat. It's an autoimmune rat model of various other autoimmune diseases, and so we create myocarditis because I have a whole myocarditis program in my laboratory that we study it. But we used it thinking that maybe we could see the brain disorder come to life in the model, and so yes, the animal, the rat model, the transgenic mouse, we did not do the behavior, unfortunately. So, but in the rat model that I was showing, that is immunized with the strep, and yes, I didn't have time to show everything we've done, but we have taken the purified IgG from the rat model that gets all the manifestations, and they were put intrathecally into the brain. That's published in Brain Behavior and Immunity just recently. And a portion of the behaviors were transferred. In other words, the animals actually had constant marble burying. They had something, it was a little bit different, but they were, of course, very OCD. So the antibodies did transfer that type of behavior.
Right.
Yes, is that what you were asking?
I was asking CSF levels of brain-derived neurotrophic factor.
Right. Well, the brain neurotrophic factor we have not studied, but I think it's a great idea, and I'm so glad you bring it up because there's so many things that, we don't know, and we all have to pitch in with our new ideas because I may not think of the brain, I had not thought about the brain neurotrophic factor, but I think that would be wonderful for us to study. But we've not tried to transfer disease with CSF.
Right.
Because I think you were asking that too.
Right.
Yeah.
Thanks.
Mi chiamo Rosario Savino, sono un neuropsichiatra infantile, lavoro nell'ASL Napoli 1. Volevo chiedere alla dottoressa Cunningham due cose. La prima, il Cunningham Panel, che cosa va a testare? Cioè gli anticorpi anti-D1, anti-D2, anti-lisoganglioside, eccetera. A me interessa molto per la clinica, chiaramente, perché vedo molti bambini e quindi ho bisogno di uno strumento diagnostico validato. La seconda domanda, volevo sapere se ci sono degli studi, se nei suoi studi e sono stati considerati il ruolo delle interleuchine. Quindi che ruolo hanno nella Pandas e nel PANS le interleuchine pro-infiammatorie, per esempio l'interleuchina 6 e l'interleuchina 1?
Patients, but one would expect that they would be highly elevated, and I actually don't know of any. Dr. Trifoletti, do you know of any studies that have been published that have shown the interleukin levels? One thing that I can say that they will interpret for you is that studies have been done with group A strep in the tonsil, and they actually lead to T helper 17 responses. And IL-17 is very well known to be pro-inflammatory and associated with extracellular bacteria like group A streptococci and helping clear out or clear the extracellular pathogen. And as that happens, the interleukin 17 comes up, but it's not yet been studied. That's something that we hope to do in the future, is actually study the blood and study a T regulatory Th17, IL-17 presence in patients with pandas. I do think that probably one of my ideas has been that in a lot of autoimmune diseases, including things like pandas, is that immune complexes are very important. And they may also complicate treatment because immune complexes do something that you probably don't realize. One of the very best immunology papers in the last 2 years in the Journal of Experimental Medicine described how antibody, when it comes up to a particular antigen, it feeds back on the germinal center and shuts it down. Well, if you have a child that has all these immune complexes, then those antibodies are not free to go back and turn off their immune response. And so the immune response just continues, like in lupus or in Sjogren's, and it continues until you can stop it and get the immune complex level down. It doesn't mean that mimicry doesn't help cause the disease, but immune complexes just continually promote inflammation. And I think it's why there are complications with the treatment. Some people get better and other people you have more trouble with.
L'altra domanda.
Cunningham Pana.
Ecco, Becconi.
Dopamine, yes, yes.
According to the treatment,
Yes. You're asking if of course, if you know that antibodies are present, then it makes it more clear that it would be an immunotherapy. You would not want to treat someone that had no antibody, that has just garden variety OCD or a Tourette's case that doesn't have it related to the immune system. So a lack of antibody suggests that maybe it's not an autoimmune, that particular case, because that's why we develop antibody assays, is so that we can understand that in those children that have the antibody levels, then they are the ones that you would want to treat with immunotherapy.
Pensi che nella pratica clinica sia necessario effettuare il test Cunningham per trattare questi pazienti? Chiedo.
Conflict of interest for me to answer this, but I, as an immunologist, and because I'm always think it's very important to know as much as you possibly can about every patient, that yes, you should perform any test that will give you more information about the child, and their condition, because it's like an autoimmune encephalitis. If you think they have autoimmune encephalitis, you're going to run anti-NMDA. Now we have the anti-dopamine receptor antibodies. It's just a different type of encephalitis, that's all.
Too expensive in Europe to perform the test.
Well, you know, I understand well, the test is performed by WeSlab, and Dr. Ako is here to answer any questions. And they actually, it's performed in our laboratory, our molecular labs in the United States, but it's through WeSlab here.
Allora, chiudiamo questa presentazione ed invitiamo la
E ho mia figlia Pandas. Ecco, abbiamo
Can you
fatto
hear me now?
i test in
I'm a
Svezia, ma mi
psychotherapist. My
chiedo
daughter has
quanto
pandas.
questo
We
ci permette di
tested
fare una diagnosi
we made the test
differenziata.
in Sweden.
Perché di fatto in allegato comunque viene detto che questi valori possono sì indicare Tourette, Pandas, però non sono così discriminanti. Per cui mi chiedevo appunto questo, dico ai fini del trattamento che cosa ci facciamo poi con il test?
Her question was about the test discriminating for the different diseases. I think it's better to answer by saying what we know about pandas that Dr. Sweeto was talking about. The pandas that's like Sydenham, Korea, is pandas, and that has elevated D2 antibody, and anything else that is more chronic does not. It will not have D2. I mean, that's what you were asking, correct? So, you know, and as far as your decision is that if you have elevated antibodies, yes, the report says, you know, we don't want to say it's diagnosing it necessarily, but that it gives you a guide with the clinical symptoms. I mean, there may be children at risk. We are still running controls, you know, to look at the control population of children. But like if it's a child that maybe has had a lot of infections, it might get elevated. So those would not be in our control group. But you have to be aware of that. Does that make sense? Okay, good.
Thank you very much. Grazie alla dottoressa Cunningham.